Welcome to company presentation by ImaginAb. My name is Plavec. I'm a Chief Business Officer. The title of the talk is PET Imaging of CD8 Cells in Cancer Patients. A few quick facts about the company. We are a clinical stage biotech company based in Los Angeles, utilizing engineered antibodies, minibodies, and cis-diabodies for the development of radiopharmaceuticals. Our lead product, CD8 ImmunoPET, for non-invasive PET imaging of CD8 cells, is currently in phase 2 and has been already broadly adopted by pharma as a universal pharmacodynamic biomarker for immunotherapies. Our platforms enable rapid to-market development of new imaging agents and radiotherapeutics. We are backed by an excellent team of advisors, including Jim Allison, a Nobel laureate, Pam Sharma, Daniel Van Hoff, Tony Ribas, and Rami Ibrahim. All of them are leaders, global leaders in immunotherapy, immuno-oncology, as well as imaging specialists and KOLs, including Anna Wu, our founder, David Manko from UPenn, and Jeff Evelhoff from Merck. Our platforms have been redesigned and improved to serve a product development. So we have a proprietary generation 2 designs for minibodies and cis-diabodies. They still retain high affinity and selective targeting properties of parent antibodies, but they have been redesigned to stabilize smaller fragments so we get uniform drug product, improve production, high titers, and no aggregation, as well as immunologically inert structures. We have an efficient process to convert full-length antibodies to minibodies or cis-diabodies. And one nice feature of these is that both formats can be made bispecific if the product requires that. We also like the PK of these proteins. Minibody about 80 kilodalton, cis-diabody 55 kilodalton. They quickly clear from blood circulation, which is good for radiopharmaceuticals, but they still retain, maintained in the circulation for long enough such that we get a high uptake. Rapid tumor penetration and high retention results in excellent tumor uptake and a very good signal-to-noise ratio. To make a product, you combine a binder, which is either minibody or cis-diabody, with a chelator. We have various chelation systems for biologics, and at the end, you add an imaging isotope or therapeutic isotope to either create an imaging product or a therapeutic product. The topic of today's talk is the CD8 ImmunoPET minibody that's been developing. We are developing it for PET imaging of CD8 cells. The minibody itself is 80 kilodalton, covalently bound to the chelator desferrioxamine, and in the final step of manufacturing, radioactive zirconium-89 is added, giving a product that has a 6-day shelf life. It has a high affinity for human CD8 alpha chain. It's immunologically inert. All the effector functions have been removed from the minibody, so there's no CDC, ADCC, or FC binding. And a half-life of 78.4 hours allows two things. One is central manufacturing, so we can ship from a central site to all the clinical sites on the continent and even between continents. And the longer half-life allows us to do serial imaging after single infusion of the tracer, which is important for some immunotherapy clinical trial designs. Why are we focusing on CD8? Now, immunotherapy, specifically checkpoint inhibitors, have really changed the landscape of cancer therapy, providing durable response and much better response than has been previously seen with chemotherapies. Now, that said, there is still a large variability in patients' responses to immunotherapy, and only a subset of patients show true clinical benefit, some in the range of 20 to 40%, meaning that we have about 80 to 60% of patients that do not benefit yet from immunotherapy. CD8 cells, CD8 T cells, or the cytotoxic T cells, have emerged as a critical immune cell mediating the efficacy of immunotherapies. There's literature out there showing that if CD8 T cells are depleted from an animal, those animals no longer show any efficacy against tumor when treated with immunotherapy. So we believe that imaging CD8 cells in the whole body of a patient could improve the effective use of approved checkpoint inhibitors and inform the development of new therapies. We have completed the phase 1 trial with our product, established safety and proof of concept for imaging. It's been published in the Journal of Nuclear Medicine, and there's a paper on the right here, an image from it. We also established the optimal dose and the optimal imaging schedule. And we use these parameters in our phase 2, which is ongoing. We were looking at the safety of repeat doses. We're assessing diagnostic performance, namely correlating CD8 PET to CD8 IHC from biopsies. And we assessing predictive performance, correlating CD8 PET with RECIST and clinical outcomes to see how the CD8 PET images could be used to provide an early prediction of the clinical benefit for the patients. We are enrolling patients with locally advanced metastatic disease that are on standard of care checkpoint inhibitors. We have enrolled 22 patients to date. And now let's look at some images. There's an image of a patient, a melanoma patient, taken at the baseline, meaning before the patient is being treated. And what immediately becomes apparent is that it's a very clean PET image. There's not a lot of background. What we see is images of normal immune organs, lymph nodes, spleen, and bone marrow, here with red arrows. We see this in every patient that we image. This patient has a tumor in its right calf, and so we can see some infiltration of T cells into that tumor lesion. The tracer clears through liver and through kidneys, and so there is a somewhat higher background in those two organs, but the background is not terrible, so that we can readily see tumor lesions, metastasis, both in livers and the kidney, and it's not an issue for the performance of the technology. Other tissues have a really low background, and so we can readily detect any kind of inflammation or immune response where the CD8 T cells may be infiltrating, and it is important to monitor also immune-related adverse events that may occur upon immunotherapy treatment. And now let's look at some tumors. The resolution of CD8 PET is really good. It does enable us to classify tumors based on CD8 cell infiltrate. This classification of tumor being hot, excluded, immunosuppressed, or cold has been described in a Nature paper that came out last year. And that classification was done using biopsy data, so immunohistochemistry data. But we see very similar patterns in CD8 PET images. So we see some tumors that are hot, showing uniform staining throughout the tumor. Here you see a melanoma. Here you see two lesions in the liver of a hepatocellular carcinoma patient. We also see excluded and immunosuppressed phenotypes. Here's an excluded melanoma tumor in a newly diagnosed patient. And here is a immunosuppressed, meaning weakly staining for CD8 PET in this large lesion found in the pelvis of this acral melanoma patient. And a cold lesion here in a lung cancer patient. Again, this is a lesion in the neck, one of the metastases, which is fully viable and highly avid on the FDG PET, but there are no CD8 cells in that lesion. By doing serial images, we can measure changes in the CD8 cell distribution that's induced by checkpoint inhibitors, or for that matter, any immunotherapy. And we can measure these in a tumor as well as in immune tissues, such as lymph nodes, bone marrow, spleen, as well as measure in the non-immune, non-tumor tissues to look for sort of ectopic sites of inflammation that may indicate adverse events. And so you're seeing here, this is a melanoma patient. I showed these images before. This is a baseline scan when the tumor is excluded. On treatment, that patient has a really good response. This is 4 weeks on pembrolizumab. This patient, acral melanoma, that was immunosuppressed at baseline, is still immunosuppressed after 4 weeks on nivolumab, and the tumor is actually larger. So this is a true progression here of a tumor. In this lung cancer patient, there is I'm pointing here out to the mediastinal lymph node that got activated after four weeks on a treatment of nivolumab. It's a very bright staining, 4- or 5-fold increase in a signal, indicating a quite strong activation of the immune system in this patient, even though the tumor infiltration itself has not yet reached the maximum. You can see heterogeneous staining at the baseline, and still heterogeneous, albeit higher staining on treatment. these observations from the phase 2 has allowed us to develop a thesis as to how CD8 PET can be used in clinical development. And some of these theses have been developed by us, others were actually suggested to us by our pharma partners who also think carefully as to how to use this for their own purposes. So one obvious application is then one can address CDL trafficking, CD8 cell trafficking by comparing baseline to on-treatment scans. One can also start comparing combination treatments to monotherapy. One can establish specific patterns that may predict clinical benefit and as well as side effects, and that's unique to the whole body imaging. You can do this with biopsies, for example, or by collecting blood for liquid biopsies. One can look at the intrapatient and intrapatient heterogeneity, and it's known that, for example, depending on the site, the metastases respond differently to treatment. So that's a very powerful technique to look at, let's say, bone metastasis or brain metastasis versus soft tissue metastasis. And one can, of course, stratify patients based on baseline scans before one starts the therapy, especially if one starts with something that's has a novel mechanism of action. One would look for the abscopal effect, or can one turn cold tumors into hot. So such data before one starts therapy would be very important. And of course, one can look at to establish optimal route, dose, and timing of administration. Here are some of the deals, some of the companies that are working with us already using this technology as a pharmacodynamic biomarker. And if you would be interested to work with us, we make the technology available under non-exclusive license, so you can incorporate CD8 imaging into your clinical trials. We also have formed a pre-competitive CD8 collaborative group, which gets exclusive access to our clinical data from our phase 1 and phase 2 studies, as well as participate with us and other members, right? We all jointly developing analysis and standardizing the use of CD8 imaging for clinical applications. So if you are using CD8 imaging in your own clinical trial, being a member of CD8 collaborative group can really empower the use for your proprietary projects. And on the platform side, we are very much interested in partnering around radiotherapeutics, specifically because we don't see ourselves only as an imaging company. We are a radiopharmaceutical company, and that's the next thing on our books to develop. We have some internal programs, but others we'd like to partner. And with this, I want to thank you for listening. If you want to contact me, we can meet at bio, or you can reach me at this email address outside of the conference. Thank you so much. Enjoy the conference. I look forward to hearing from you.